We were discussing this chapter number 11, biotechnology, principle and its processes. In this chapter, in the previous session, we have learned regarding the definition of biotechnology, what are the principles of biotechnology, and the steps involved into a genetic engineering. But when we are discussing such kind of an engineering, genetic engineering, it requires a different tools. Without the tools, the process does not happen, or it is not possible to convey the process without these tools. So today we have to discuss the tools which are required in this genetic engineering process. So let us start with the first tools which are involved into a genetic engineering. Okay? Tools which is required into a genetic engineering. So what are the different tools are required in the genetic engineering? The first tool which is going to be required in the genetic engineering is the enzyme. Which enzyme? Restriction enzyme. This restriction enzyme, they are responsible for cut the DNA from specific locations. So this is the one thing that we require. Second, we also require the ligase enzymes. Ligase enzyme, they are used to ligate it, join it, and to form the recombinant DNA. It also requires the polymerase enzyme. Polymerase enzyme that makes the multiple copies, the millions of copies are going to be formed. That is done with the help of this polymerase enzyme. The next thing which is going to be required is the cloning vector. The next tools what is going to be required is the host cell, in which we have to introduce this recombinant DNA which is going to be formed. Okay, so these are the different tools they are going to be required into a genetic engineering processes. We have to discuss the characteristic of each tool and how they are working in this chapter. So let us start with the first one. The first tool that is going to be required in the genetic engineering, it is called as restriction enzymes. So first of all, we are going to start with this enzyme called as restriction enzymes. And then after, we will discuss the other tools which are required in the genetic engineering. Once we understood regarding this tool, then it is easier for you to understand the process of genetic engineering. So see that in the year 1963, the two enzymes have been isolated and have been recognized, which are restricting the growth of bacteriophage in the E. coli bacteria. So we found that which two enzymes are that. From these two enzymes, one enzyme was adding the methyl group to the DNA molecule, and it is restricting the growth of bacteriophage in the E. coli. And the other one is this enzyme, this is called as restriction enzymes. So what are called as the restriction enzymes? Restriction enzymes, they are meant to cut the DNA from specific location. Okay, they cut the DNA from the specific location. Okay, clear? So specific location, that means here it identifies how it decides the specific location. It identifies the base sequence. Okay, they identify the specific base sequence, and this sequence from where it will cut, this is called as the recognition sequence. What it is called as recognition sequence. So first of all, it will recognize from where to cut. So for that, it will look after the specific bases. Okay, and this base for which it is looking after, this is called as recognition sequences. With the help of this recognition sequences, it identify and cut at that from that specific locations. So these are the enzymes which are going to be useful to cut a specific portion of a DNA. The first restriction enzyme that was discovered is Hind2. This was the first restriction enzyme that was discovered, Hind2. Okay, when we are looking after the working of this Hind2 enzyme, Hind2 restriction enzymes, then we found that always it cut from specific sequences. Always it is cutting from the specific sequences. So it is recognizing the 6 base sequences and it is cutting from that portion always. Clear? So this is what usually happens in the case of this enzyme that was called as HIND2. So the first restriction enzyme that was discovered was the HIND2 enzymes. And then the journey goes on. And now currently we have more than 900 restriction enzymes we have. And these 900 restriction enzymes we have isolated from 230 different strains of microorganism, from which we have isolated 900 restriction enzymes. Currently we have a different 900 restriction enzymes available. They cut the DNA from the specific location or from the recognition sequences. Okay, and that was isolated from the 230 strains. Now when we have discovered so much restriction enzyme, now there is a problem comes regarding their nomenclature or how to give their name. Clear? And so here the specific system has been developed. I will explain you the naming method or the nomenclature method for restriction enzymes. Let's see that this is one of the example of a restriction enzyme, EcoR1. Clear? It is not I, it is 1. Clear? EcoR1. So what this EcoR1? This is the name of restriction enzyme. It is a restriction enzyme. Clear? How it is going to be named? Okay? So here this first letter, it indicates that it is the genus of a bacteria from which it was isolated. So here the E means Escherichia. So it is the genus. This is the enzyme which was isolated from, you can say that bacterial strain Escherichia coli RY13. Okay, from which it was isolated? It was isolated from the bacteria called as Escherichia coli RY13. So here the first letter of this, it indicates the first letter of a genus. So this is an E. The next two letters, they are the letters from the species, CO. Clear? So this is the first letter, second letter and the third letter. So the first letter is the first letter of a genus. Second and the third letter, they are the letters from the species. Now afterwards there is an R. The R indicates the strain from which it was discovered. Clear? So the next letter is the strain from which it was discovered. And the next last one is the Roman letter. Okay? The Roman letter indicates the order in which it was isolated. Which was the first, which was the second, which was the third, etc. It is according to order of discovery, they are going to be given a letter in a Roman letters. Clear? So this is a way how the restriction enzymes are named. So I am again repeating, what is this EcoR1? R1. It is not I, again repeating. EcoR1, it is a restriction enzyme. How this name is given? There is a specific way. The first letter indicates a name of genus, first letter of genus. The second letter, CO, that indicates the name of a species. The next letter that indicates the name of a strain from which it was discovered. Here it was discovered from the strain RY13 of E. coli. And the next is a Roman letter which indicates the order of discovery of this enzyme. So this is how the nomenclature is given, because now currently we have more than 900 restriction enzymes. So it is difficult to give its name and remember. So here a specific kind of a nomenclature system has been developed. Now when you see that this restriction enzyme, they are cutting from the specific sequences. But if you see the characteristic of this enzyme according to international classification, then you can say that the restriction enzyme, they falls broadly in the category called as nuclease enzyme. Which category of enzyme? That means nuclease enzyme. Nuclease enzyme means the enzymes which are capable of digesting the nucleic acid. They are called as nuclease enzymes. Okay? And the nuclease enzyme can be of two types. There are two types of a nuclease enzyme. Which are the two types of a nuclease enzyme? Exonuclease, and the second one is called as endonuclease. Clear? So the restriction enzyme can be of two types. Exonuclease, and the second one is called as an endonuclease. So what is called as an exonuclease? Exonuclease, that means suppose here it is a DNA sequences. It is the structure of a DNA. The one chain is running from 3' to, sorry, I am writing over here 5' to 3'. The another chain is running from 3' to a 5' because they are anti-parallel and they are twisting around this single axis. So what is called as an exonuclease? Exo means outside. So here they are of category nucleus, means able to cut the DNA molecule or can able to digest this DNA molecule. But here the exonuclease, they cut the DNA from the end. Exo means outside. So it cut the DNA from the end, either of the end, they cut it. So this is called as exonuclease enzyme. And what is called as an endonuclease? Endonuclease enzyme, they do not cut from the end, but they cut from within the sequences. Like they cut from over here or any specific site, but they are not cutting the DNA from the end, they cut within the sequences of the base pair. Okay, तो एक सिंपल लैंग्वेज में कहिए तो एक्सोन्यूक्लियस दे कट द डीएनए फ्रॉम द एंड, बे छेड़ा परથી कट करे, एंड द एंडोन्यूक्लियस it cut the DNA within the sequences, means એ વચ્ચે ગમે ત્યાંથી એડ કરે, ગમે ત્યાં means from the recognition sequences or specific location they will cut, પણ એ વચ્ચેની કોઈ જગ્યા હોય. Clear? So this is called as endonuclease. Okay, so what is a restriction enzyme? Restriction enzymes are of category nuclease enzymes. They can be of two types, restriction enzyme, exonuclease and the endonuclease. Exonuclease, they cut the DNA from the end, and the endonuclease, they cut the DNA within the sequences. So these are the category of an enzyme. Now you know that they cut the DNA from the specific sequences or specific locations, what we are calling as recognition sequences. So see how the cutting can occur. So suppose it's a structure of DNA. One chain is running from 3' to 5'. The other one is running from 5' to a 3'. Now what happens? Now they cut it. So what happens when they are cutting it? Like here, such kind of a cut occurs. But when such kind of a cut occurs, here the ends which are going to be produced, they are the perfect one. So here we are going as a blunt ends are going to be produced. I am showing the another characteristic. Like suppose assume that this is the another DNA molecule. Okay. Here also the chain is running from the 3' to a 5' and 5' to a 3'. Here the cutting occurs like this. Okay. Here the cutting occurs perfectly, but here the cutting occurs like this. I am joining the remaining one. See, so here the cut occurs like this, not a perfect cut occurs, but here if you see that the ends which are going to be produced, they are not a sharp, they are not a perfect one. So here the sticky ends are going to be produced. What kind of the ends are going to be produced? Sticky ends, and these are called as a blunt end. In simple language, if I want to show you that, like suppose this is a DNA, two chains of a DNA. Suppose the cut occurs like this. This is called as a blunt. But if the cut occurs like this, like suppose this DNA, here if you see, the cut occurs, improper cutting occurs, like see, then this is called a sticky end. Okay? So both kind of the cutting can occur, blunt and the sticky end. But for genetic engineering, we require sticky ends. Blunt ends are not useful in genetic engineering. For genetic engineering, the sticky ends are required. भले गमे त्यादी cut करे, whether it is from the end or whether from the within the sequences. But the blunt ends are not going to be utilized in the genetic engineering. The sticky ends are going to be utilized in the genetic engineering. Okay, clear? Now, the point that comes to that, here it cuts from cut from the specific sequences. So let's see what is the specific sequences. What kind of the sequences it will recognize? It will recognize the palindromic sequences. What kind of the sequences it will recognize? The restriction enzyme, it will recognize the palindromic sequences. Okay, which is also called as a recognition sequence. Palindromic sequences means the sequences which can be read same in both forward and the backward direction. This is called as palindromic sequences, as we have discussed in the chapter number 6. Like I will give you example. Malayalam. This is the palindromic word. If you read from this direction or this direction, it is same. Or if you see the another example, the rotted or madam, etc. There are many examples which are read in the same. Okay, in the forward and the backward direction, these are called as palindromic sequences. So it will recognize these palindromic sequences and cut it. So let's see in the terms of DNA, what is a palindromic sequences? I am just giving you one example, but there are many possibilities. And for easiness, I am drawing just two chains like this, so you can easily recognize. So here this chain is running from 3' to 5'. The another one is running from the 5' to 3'. So what is the palindromic sequences? I am showing you G, A, T, T, C. Suppose this is a sequence which exists on the 3' to 5' chain. Now you can also able to write the complementary base pair. So write down C ni sa mein G, T ni sa mein A, T ni sa mein A, T ni sa mein T, T, C. Okay, so now you read in this direction 3' to 5' G, A, T, T, C. Now read in this direction this way. Okay, clear? So you can read the G, A, T, T, C. Okay, so when you read in this opposite directions, then it is read as the same. Such kind of a sequences, they are called as palindromic sequences. Okay, and here it will cut from the specific side. Like for an example, in this case, it is cutting from this side between the G and A, between the G and A. So what kind of the structures is going to be produced? Such kind of a structure is going to be produced. Like say G. Now here it's a cut. So what it becomes? A, T, T, C. Now what is the second cut? Like this. You know that where it cut? It cut between the G and A. So what is the sequence? C, T, T, A, G. Clear? This is a sequence. So if you see that, the cut occurs in this way, and thus it produces the sticky ends. And these sticky ends are useful in the formation of recombinant DNA. Now these sticky ends can able to combine with another DNA molecule. Okay, it can able to join with another DNA molecule with the help of a ligase enzyme. How it occurs, that we will see later on. But here the function of this restriction enzyme is to cut the DNA from the specific site, specific locations by recognizing the recognition sequence, and usually it is a palindromic sequences. The end which are going to be produced or useful in the genetic engineering are the sticky ends. Blunt ends are not going to be utilized in this genetic engineering. Okay, so now we are moving ahead. Once this is going to be produced, these ends will be joined with another DNA molecule, and we can able to form the recombinant DNA. Okay, but now what we have to do? Once the DNA has been cut, let DNA na tukda thai gaya, it is converted into a pieces. Quick nano hase, quick moto hase, someone is a medium in size. This pieces has to be separated out, because from this pieces or the fragments we have to utilize either one or two to form the recombinant DNA molecule. But that tukda apne use nahi karwana. We are utilizing it to form a recombinant DNA in a specific fragment. So what we will do, once we cut it, then we are applying the next step, what you can say that separation. So we are moving ahead in this one. Once the fragments are going to be formed by restriction enzymes, then the separation occurs. Separation of this fragment occurs. Here these fragments are of different size, larger size, smaller size, medium size, different size of fragments are present. So we are separating these fragments, and which technique we are using it? We are using a technique called as gel electrophoresis. Which technique we are using it? We are using the gel electrophoresis technique. So what is a gel electrophoresis technique? In the gel electrophoresis technique, there is a one stationary medium. Okay, like suppose that this is the stationary medium. Here it is a gel is acting as a stationary medium. Okay, usually we are using the gel. I am telling you the name. It is the agarose gel. Which gel is obtained? The agarose gel. What is agarose gel? If you see this agarose gel chemically, then chemically it is a natural polysaccharide. It is a natural polysaccharide which is obtained from seaweed. Okay, this is a agarose gel. Okay, clear? So what is agarose gel? Here the agarose gel is used as a stationary phase. That's why we are calling it as gel electrophoresis. And this agarose gel, it is a chemical, it's a natural polysaccharide and obtained from the seaweed. Okay, clear? It is a kind of a marine organism from which we are obtaining. Now what happens? Here, whatever the fragments are there, we are keeping these fragments over here of different sizes. These fragments are kept over here. Okay. Now you know that they are the DNA fragments. DNA is the negative charged. You know that DNA is the negative charged molecule. So here we are applying the current. What we are applying? We are applying the current, electricity. And the electricity also has a negative charge. This DNA also has a negative charge. This electricity also has a negative charge. So we are applying the fixed current. Rubber check, clear? So now what happens? This is a negative charge, this is a negative charge. So what happens when the two negative charges are present nearer to each other? A repulsion force occurs. And due to this repulsion force, the molecules are going ahead. Okay, they are going to be separated. Like see, this kind of a separations you can find over here. This moves. Okay, it moves towards its opposite charge. Which is the opposite charge? Here the opposite charge is the anode. Means it is a positively charged. Clear? The DNA is the negative charge. Here we are applying the current. So repulsion force occurs because both are same charge and they move. Move towards what? They move towards the anode. Okay? So such kind of a procedure is called as electrophoresis. As we are applying the current over here or these molecules, we are applying the current. So that's why this is called as gel electrophoresis. Here the DNA molecules, they are separated based on the size. The smaller DNA fragment or smaller, jitno nano ase etlo dur jase. Like you see that, if you see the different sizes of ball, okay, now if you kick them, okay, you are kicking them with the same force. Which ball goes very farther? The ball which is a very small, that will go very farther. Je nano ball ase is sauthi dur jase, because every time you are applying the same kick. Clear? Okay, and je moto ase in najik jase, dur jase but najik jase. So here the separation of DNA fragments, that based on the size of the DNA fragments. Based on the size, the electrophoresis separates this DNA molecule. Okay, once the DNA molecule that is going to be separated, okay, you want to pick a piece which is required to make the recombinant DNA. So how will you do this? Before picking up, you should able to view it. Usually these DNA fragments which are separated on this gel electrophoresis are not visible through a naked eyes. DNA fragments are very small. We are not able to see it through a naked eyes. So what we have to use? We have to apply a special kind of a stain. And here the stain which is going to be used is ethidium bromide. Which stain we are applying? We are applying the stain called as ethidium bromide. When we are applying this ethidium bromide, the color of this DNA, it converts into a brown. Okay, it converts into a brown color. But still, it is not seen under a visible light. The normal sunlight under this brown color cannot be seen. So here, after applying this stain, ethidium bromide, we have to observe under the UV light. When we observe under the UV light, we can see the brown or the brown-orange color stains. Okay, clear? A brown-orange color stain is observed, and they are considered as the fragments of a DNA. Remember, this DNA cannot be observed through a naked eyes. Require a special kind of a staining chemical like ethidium bromide, and then we have to observe under the UV light. Then we can see the brown orange color, the stain of a DNA fragments. Even it cannot be seen under the visible light. Okay, so this is what happens, how we can see it. Now once we observe it, like when we observe under a UV light, we can see the such kind of a fragments of DNA separated on the agarose gel. Now what we will do, suppose I need this fragment, so I will cut this piece of a DNA, I will cut this gel, clear? And from this, they are going to be, this DNA molecule is going to be removed out, or it's going to be separated out, because here it's a DNA and other component, so we are separating out after cutting it. So after we are observing, we are cut the portion of a gel, then we are removing out the DNA, from it, because here it's a mixture of gel and the DNA. Barabar છે? Clear? So we are removing that, and then we are going for purification of this DNA. Okay? कोई भी impurity है, then we are removing this impurity by removing the DNA and purification of a DNA. Okay? And that process, removing the DNA and purification of DNA, this process is called as elution. What is called as elution? And thus you get a purified DNA molecule, which is used to join with another DNA molecule, and that is also obtained by the same way. Okay, you cut it with the help of restriction enzyme, then the fragments are going to be separated by gel electrophoresis, then it is visualized by application of a stent, ethidium bromide and the UV light, then the desired DNA is going to be cut, it is removed, and then it is purified, and thus you get the two different DNA molecules. In the next session we will see that how they are used or we can join it. So here we have discussed the first tool that is required in the genetic engineering, and this is called as restriction enzyme. It is an important tool. It is also important from examination point of view. So students, learn it thoroughly, keep learning, keep watching, and like and subscribe this channel. Thank you for watching.